Hello and welcome to another screencast with Life at Phuket Pals. This is teacher Alex, and today we will have a look at the GT Science topic, evolution, with a specific focus on cladograms. So what is a cladogram, first of all? A cladogram is a diagram used to represent a relationship between organisms of animals, called phylogeny. A cladogram is used to show how groups of these organisms are evolutionarily related to each other and usually show their most common ancestor. A cladogram can be very simple, comparing only 2 or 3 groups of organisms, or it can be enormously complex and contain all of the known life forms. As we can see down here we have a relatively simple diagram, simple cladogram of a few groups of organisms. The cladogram itself gets its name from the word clade or clades, and a clade is a group of living organisms and the common ancestor they are derived from. So there is another video that investigates evolution and evidence for evolution and common ancestry. So if you want to know more about common ancestors and what this exactly means, have a look at that lesson. Down here, quick explanation of what an ancestor is. An ancestor is an early type of animal or plant from which others have evolved. So for example, if we look at this cladogram down here, and we have the mouse and the chimp, then their common ancestor is where the 2 lines meet. And the common ancestor of a mouse and a chimp had mammary glands and fur. So the mouse and the chimp are a clade in this cladogram. This is, as I said, a relatively simple cladogram. It goes to very complex. For example, here we can see more or less the complete tree of life as far as we know it, and that for sure is a very complex cladogram. It really depends on the researcher or you, what you want to investigate, and the relations between organisms you want to look at. So, what are key features of a cladogram? First of all, a cladogram shows organisms or groups that are studied and that are of interest for you. Again, it's up to the scientist, it's up to you what kind of organisms you want to study. So you can make a simple or a complex cladogram, depending on your field of study and your interest. The lines in a cladogram represent evolutionary time. So the further back we go in a cladogram, the further we go back in time. The root is the base, the initial ancestor, the initial common ancestor of all the organisms in your cladogram. And that's the incoming line, and it originates usually from a larger clade. The nodes, which we can see here, for example, this is a node. Another node would be here. Each node corresponds to a hypothetical common ancestor that gave rise to these 2 new species. And these 2 species then, including the common ancestor, are a clade. The outgroup is the most distantly related species in the cladogram, which usually functions as a point of comparison or reference for the other groups or for the other clades. It's the most distantly related group or species. The outgroup in this cladogram we have down here. C is the outgroup because it is on that node which is furthest away from our other groups. And again, a clade is a common ancestor and all of its descendants. Node and all of its branches and connections. Another important feature is that in a cladogram, on a node, lines always diverge. So we come from the past, go towards the present, and evolutionary time, we go that way. Here is probably the present. And lines always diverge in a cladogram. They never converge in a cladogram. They don't come together. We don't see that. Two species never fuse into one species. Once two species exist, they continue to exist until they eventually go extinct, but they will not fuse into a single species. So lines always diverge, they never converge. All right, let's construct a cladogram. How do we do that? Cladograms can be constructed based on either a comparison of morphological or structural features of organisms or molecular evidence. We will only go over one example here using structural features. So structural features were historically used more often to construct cladograms because we didn't know in the past anything about molecular biology or didn't have the means to investigate that in the past. Nowadays, phylogenists and people who construct cladograms and are interested in the science usually use molecular evidence, sometimes in combination with structural features, to create their cladograms. So we have a couple of features here in our table and a couple of organisms or groups of organisms. We have fish, amphibians, reptiles, birds, rodents, and primates. And the features we are looking at are vertebrae, the vertebrates, the backbone, four limbs, the amniotic egg, a certain type of egg that has an additional structure in it, hair, fur, and eggs with shells. So, what do fish have of this list? Fish have a backbone. But fish don't have four limbs, they have fins, they don't have amniotic egg, they don't have hair or fur, and they don't have eggs with shells. What about amphibians? Amphibians have vertebrates. Yes. Amphibians have four limbs. Think about a frog. A frog has four legs. Correct. Does a frog have an amniotic egg? No. Does it have hair or fur? No. Does it have eggs with shells? Nope. Reptiles. Reptiles have vertebrae. Yes. Do they have four limbs? Yes. Do they have amniotic eggs? Yes. Do they have fur? No. Do they lay eggs with shells? Yes. What about birds? Birds have vertebrae. Yes. Four limbs. Yes. Two legs and two wings. Amniotic egg. Yes. Hair and fur. No. Eggs with shells. Yes. Rodents like mice or rats have vertebrates. Yes. Four limbs. Yes. Amniotic egg. Yes. Hair and fur. Yes. Eggs with shells. No. Primates. Vertebrate. Four limbs. Amniotic egg. Hair and fur. Eggs with shells. No. So let's construct this cladogram. We draw our base. And now we can construct the nodes. Our node that is probably down here is where our outgroup goes off, which is in this case the fish. And the feature that we have down here are vertebrae. All of our organisms in this example have vertebrae. Our next group, our next node, amphibians. Have vertebrates, so they are included in this clade. Our next feature, 4 limbs. And our next is the amniotic egg. Who has amniotic eggs? All 4 remaining. So they all belong to that clade. So we somehow need to connect the next 4 organisms, that their lines all converge towards this point in the past, to this common ancestor that had amniotic eggs. Problem is, however, only 2 of the 4 have hair and fur, which are the rodents and primates, and the other 2, birds and reptiles, have eggs with shells. So that means we need to split that this way. So here we can put the reptiles and birds, and what do we have here? Their common ancestor had eggs with shells. And then we have another common ancestor of the rodents and the primates, who had hair or fur. And we have constructed our cladogram. We can play around with that a little bit and see if we can identify a few clades. We can identify the clade of organisms that have hair or fur. This is our clade, including the node, the common ancestor. That is one clade. Another clade would be the clade of reptiles and birds. They share eggs with shells. However, we can put all 4 into one clade that unifies them. That clade would be the clade with the amniotic egg. So here we have one clade in which all 4 groups, reptiles, birds, rodents and primates, are summarized in. We could include the amphibians by adding 4 limbs. What else can we say about this cladogram? Our fish are the outgroup, And down here we have our root, which could be all animals. Alright, so much to constructing a cladogram. It is important when reading cladograms or constructing cladograms to understand that we can swap groups around as long as they connect to the same nodes. These two cladograms we see on the right here are the same. We can see that they're different in a way how we draw the lines. That doesn't really matter how we do that. The more the difference that is probably more important here is when we look at AB here, we see that this is swapped, B and A. The important thing is that, again, we have to look at the lines and the nodes, and we can see that the node here and here is the same in both cases. So these two cladograms are exactly the same. A and B in both examples have the same common ancestor. Alright, let's have a look at some questions. In the following cladogram, which groups are most closely related? Beetles, wasps, bees, ants, butterflies, moths, and flies. Beetles and ants, flies and beetles, moths and flies. Flies, beetles and ants. Beetles and ants. We have a node here, we have a node here, and we have a node here. Beetles and ants not very closely related. Moths and flies are closely related. They have the same common ancestor. We go back in time, we don't have to go back far, and the two lines will meet. So the correct answer here is C, moths and flies. All right, next question. You are making a cladogram of fruit. You have 4 fruits, a banana, an orange, and a red apple, and a green apple. Important here, on appearance only. Which of these is an outgroup and which two belong to the node furthest from the start of the diagram, furthest from the root? The orange is the outgroup, and the two that belong to the group furthest away from the node, banana and red apple, or banana outgroup, orange and red apple, or banana outgroup, red apple and green apple. Have a quick think. If you need more time, you can pause the video. The correct answer here is C again. Why C? Well, first, based on appearance, so based on shape. A banana has a completely different shape than oranges and apples. Apples and oranges have more or less the same shape. They're round, bananas are oblonged. Now, why red apple and green apple and not the orange? Again, red apple and green apple are in shape and appearance very, very similar. They are both apples, whereas an orange is slightly different. Skin is more rough, size might differ. So red and green apple are furthest away from the node. If we would construct this cladogram, it would roundabout look like this. We have our banana as an outgroup, Then the orange comes in here. And here we have red apple and green apple. The cladogram was created using which type of evidence for common ancestry? The fossil record, biogeography, molecular homologies, or homologous structures? This is a tough question. We did not discuss this in detail in this screencast, but if you want to know a bit more about how to solve a question like this, you can watch the lesson on evidence for common descent, evidence for common ancestry, another evolutionary lesson. The correct answer in this case is homologous structures. Anchors of roots, vascular tissues, seeds, and flowers are all structures which we find in these organisms. Flowers can look very different, seeds can look very different, but the underlying structures of seeds and flowers in all these plants that have them are the same, and this means they go back to a common ancestor. We could very well base a cladogram on molecular homologies as well. We could as well base it on fossil record and biogeography, but in this case, these are homologous structures. Again, all these as evidence for evolution and common ancestry are discussed in another lesson. According to the cladogram, which two species are most closely related? Sharks and amphibians, crocodiles and birds, fish and crocodiles, rodents and crocodiles. Have a quick think. The answer comes now. You can pause the video. The correct answer is crocodiles and birds. B. Another answer that you might have fallen for is answer D eventually, which is not correct. We can see in answer D, rodents and crocodiles are next to each other up here, but their common ancestor going back in time is down here with the amniotic egg. So it's quite a bit of a way to get down there. Much closer related in this case are the crocodile and the bird. Their common ancestor is directly here with eggs and shells. Last question. We have 4 cladograms here. 3 of the 4 cladograms below are the same. Which one is different? Again, in our last slide, I said that in a cladogram, the arrangement of the groups on top doesn't really matter as long as the lines and nodes are the same. So what we are looking for is the nodes. The top node, the highest node in our evolutionary history, in our cladogram here, is DE. The second node is C with D and E. Then the third node is B connecting to C, D and E. And the last node is the outgroup A. Let's have a look at the rest. A connected to the rest, that's fine. A connected to the rest, A connected to the rest. So that's the same in every cladogram. Now, second node is B. That is correct. B is connected to C, D, and E. That's so far fine. Here, our second node is B as well. Again, we can swap. It doesn't really matter. So that's still fine. And here our second node is B as well. That is still fine. Our third node, C, connected to D and E. C connected to D and E. That is still the same as our first cladogram. Our third node is now here. It is C connected to D and E. That is still correct. Here our third node is still C, and in this case E, but we can see when we look at the last node, ED, DE correct, DE correct, but in this case it is D and C. This makes our fourth cladogram different from the rest. All other three cladograms, cladogram 1, 2, and 3, are exactly the same. Alright, that's it on the topic evolution and cladograms. I hope the topic of cladograms, these diagrams, are now a bit more clear to you. I hope you've learned how to construct them using structural features or morphologies, and know how to read and interpret them. Hope to see you next time. This was Teacher Alex with GD Live at PALS. Have a great day.